asks about stomach stapling. Just in case you do not remember me talking about this before, that's the idea literally that you get staples or some other physical thing put on your stomach so that it cannot stretch. So it prevents expansion or stretching in your stomach. Your stomach is actually a muscular organ that can stretch. If I cannot stretch it, then less food enters. And that is the basic idea behind stomach stapling. The key point there is that I want you to remember that your stomach is not made out of fat. And a lot of people have the misconception that because from the outside, many people have fat on the area of their body where their stomach is. Remember, your stomach is way on the inside. There are a lot of layers of tissue between your skin and your stomach. And fat is on the outside of the organ. The organ itself is not made out of fat. The second question is asking you about stomach acid entering the esophagus. So that means that it's coming up. So when you look at your stomach, this stretchy little bag, there's a valve at the top and a valve at the bottom. Valves are muscles, circular muscles, that can open and close to allow food to pass through. The valve that is at the top is the one that we would be focusing on here. It is the cardiac or esophageal valve. If I had been asking about the valve at the bottom, this valve is called the pyloric valve. Now the third question actually has a point where there's more than one answer that would be, I think, correct. The third question asks, if the pH increased, what could be the cause? So there's two things that can make the pH increase. Either I add base or I take away acid. Either one of them could make the pH increase. So for point number one, either my gastric gland produced less acid, or my gastric gland produced more base, and in this case the specific base is bicarbonate. So the glands that are in your stomach produce a whole bunch of substances. We made a list of them last week. Acid is one of them. A base is one of the others. So either of those scenarios could result in a higher pH. Number two asks, which substance would no longer work in these conditions? So one of the things that your stomach is well known for is how it digests proteins. And to do that, pepsin, which is an enzyme, requires a low pH to be activated. Without a low pH, it remains in its inactive form, which is called pepsinogen. So the result, or the outcome or impact on digestion, would be less protein digestion. Notice I used the word less and not none, and that's because this is not the only place that proteins are digested. That's just the main thing that the stomach does. Would anyone like to ask any questions about either of those three things? So these are very similar to exam or quiz type questions that I would ask you, especially question number 3, where I give you a scenario, ask you why it happened and what would be the result of it. That is a very typical test-like question. I don't even know if I could make it even more obvious than that. I will ask you that, maybe not in those exact words, but I'll ask you that again. So what I'll do then is I'll bring back up the slide that had a picture of a gastric gland with all of the things that are part of it, just in case you forgot or you weren't here so that you can see. And then we'll continue on today talking about your small and large intestines, and then hopefully about your pancreas if we have time. So just in case anybody was feeling lost on that third question, I want to remind you this was the last slide I showed you about your stomach that showed you inside each of your gastric glands what kind of substances were produced. What I did say was you didn't have to know specifically in which order all of those cells go. It was really the substances themselves and not where specifically in the gastric gland they're coming from that was important to me. In addition to knowing which substances are secreted, you need to know what they do. So for example, you need to know that mucus protects the lining. You need to know that bicarbonate is a base, that gastric acid, of course, is an acid. You also really need to know about pepsin. That's the enzyme that digests proteins. It's pepsin when it's active. It's pepsin-ogen when it's inactive. And this is the one that requires a low pH to function. Now, did anyone have anything that they want to ask about the substances that are produced in your gastric glands? So today we are going to talk more specifically about your intestines. And we'll start with your small intestine. We'll just make this a little bit off to the side. Your small intestine has two major functions. One of them is to complete chemical digestion. So if we look at the other organs we've talked about so far, so all of them have done a little bit of chemical digestion. This is where it ends. So your small intestine has as one of its functions to complete the chemical digestion of all the macronutrients. That means carbs, fats, proteins, and nucleic acids if we include them. The second major function of your small intestine is to absorb the end products of chemical digestion into the bloodstream. So I'll just talk about the functions a little bit more, and then I'll talk about the sections of your small intestine. Knowing the function of your small intestine is great, but a lot of the time the way questions are worded is in the sense of, hey, this didn't work. Which structure are we talking about? And so that's what you could anticipate for a lot of the sections in this chapter. So if, let's say, a person had unabsorbed or undigested food coming out of their body, who could be the culprit? Well, very probably the small intestine. The small intestine is supposed to complete chemical digestion, and it is supposed to absorb all of the end products of chemical digestion into the bloodstream. So if food is left undigested, or digested food wasn't absorbed, your small intestine would be the place we would assume that the malfunction has happened. Now, is that always the answer? Of course not. There are lots of systems in your body that work together. But when we're focusing on the digestive system, that would probably be the case. The second part of this is about the sections of the small intestine. There are three of them. Their names are here. The duodenum, the jejunum, and the ileum. And I've mentioned those previously. The one that we'll focus on really the most is this guy. It is the first 20 to 50 centimeters. It really depends on how big the person is as to how long the duodenum actually is. I'm not going to ask you how many centimeters long it is. I simply want you to know that it's the first section. This is the section where your stomach and your intestine are connected. So specifically about the duodenum, this is where chyme, which is the mixture of food, etc., that came from your stomach, and fluids from a couple of your accessory organs mixed together. So we'll talk about your pancreas today, maybe we'll get to gallbladder, but those are two accessory organs. And in this spot, in the duodenum, what you ate, so your food, meets the substances that come out of those two accessory organs. So that's why the duodenum is such an important part of the small intestine, because that's where your food is finally going to meet up with a lot of the enzymes that will digest it. This mixture moves really slowly. I remember one of the courses I took in university was about nutrition, and we were always talking about intestinal transit time, how long it took for things to go through our intestines. And it's very slowly. Typically, at least 12, sometimes more like 24 hours before something that you've eaten is fully digested and moves all the way through your intestines. Now, that time could be reduced if what you're eating is something that your body is not able to digest. So, for example, if you are lactose intolerant and you drink a bunch of milk, it is probably going to come out fairly quickly because no digestion of it is going to be happening in your intestine. That's more for your stomach than it is for your intestine. Because your stomach is a muscle, right? And your stomach, right as you eat and within about 30 minutes of it, is churning the food up, like is really contracting hard. And so you don't want to do any sort of physical activity because your muscle's already working as hard as it can there. This intestine digestion process that's happening, you can be swimming while your intestines are at work. It's really more for your stomach. So the mixture moves really slowly down your jejunum and your ileum, and the whole time, all the while, chemical digestion and absorption are happening. Those are the two things that occur in your small intestine. It's possible that we could even make a case for physical digestion, but at that point, your food is basically liquid because your stomach did that work for you. As the mixture moves along your intestine, it is moving because of peristalsis, the same process that occurs in your esophagus. So when you look at intestines, they're not just from top to bottom. They're wound around, up and down. And so food is not moving because of gravity. Food is moving because circular muscles are contracting and pushing the food along down the tube. Your small intestine also has its own enzymes. And so your small intestine is a place where things can meet, but also a place where it's producing its own enzymes to digest things. So I'll just pause here. So in terms of anatomy of your small intestine, it would be important to me that if I showed you an intestine with sections on it, you could show me that the first was the duodenum, the second was the jejunum, and the third was the ileum, but not necessarily exactly how much of them are each of them. The other thing that would be important to me that you recognize is that the duodenum is where things are meeting up. So we're going to talk about the inside of your small intestine now and what is happening there. So your small intestine is one of many places in your body that has a surface area that's modified in such a way to enhance its function. And so you might have talked about this, or you might remember talking about this in Science 10, when you talked about how surface area and volume were related to each other. Other places in your body that have a surface area that has been modified to enhance absorption would be like your lungs, how they have all of those tiny little sacs at the end of each of the tubes. But the inside surface of your small intestine has three things going on. The first thing are wrinkles. So if I was to look at the inner lining of your small intestine and draw what I see, what I would see first is that the inner lining isn't flat. It's got little bumps on it. Those are the wrinkles. The wrinkles increase the surface area of your small intestine about 3 times. And when I say increase, I mean versus having just a totally flat surface. So the inside surface of your intestine has wrinkles. The second thing that your small intestines have are millions, and there are millions of these. They look like little fingers. Sometimes people say they look like the bristles on a hairbrush. Whatever you want to say they look like, they are little projections called villi. And so if I add to my picture of the internal surface of your small intestine, I would see these little finger-like projections all along the surface of it. Those are the villi. Now those villi increase the surface area 10 times further. And again, we're comparing this to if it was a totally flat surface. Now, the villi have some internal structures of their own. Inside the villi, you find blood vessels. And so if I was to take one of those little villi and sort of blow it up and make it bigger, although I'll do that without writing all over these words. If I was to take a villi or a villus, when I'm talking about just one of them, and look inside of it, here's the villus, and I would see inside of it, intertwined with each other, blood vessels. Now I drew it very simply here. I showed one red and one blue blood vessel. And then what looks like kind of a mishmash at the top. And so I'll just explain a little bit about this to you now. We'll talk about blood more specifically when we do Chapter 8. Generally, when blood vessels are drawn as blue, we're talking about a vein. The reason we would draw them as blue is because veins generally carry deoxygenated blood, blood that doesn't have oxygen. When they're red, we would call them an artery. And the place where veins and arteries meet are called capillaries. Those are really tiny little blood vessels, and they usually end up forming like a big web or a big net. So that's what the blood vessels are like on the inside of your villi. Just so you're aware, blood generally travels from an artery to a capillary onto a vein. That's the direction of blood flow. So inside each of those finger-like projections, that's where you find blood vessels. They are what are taking the end products of digestion. So by end products, we mean the digested food. Macromolecules that have been completely broken down to their smallest elements. The way that food travels from your intestine to the blood vessels is either by diffusion or active transport. It depends which mineral we're talking about or which nutrient. So what we're saying is that food, which is on this side, crosses the lining, goes into the other side, goes through, gets into the blood vessels. Does anyone want to ask any questions about villus or what's happening? I'm just going to write down, when it is singular, it is called a villus. Villi is plural. Got it. The third thing that your small intestine has is what are called microvilli. So when I look at the inside of my intestine, it looks like all of these projections have little hairs on them. I'm not going to draw them on every single one because that would take forever. But what it really looks like are little finger-like projections that are hairy. That's the appearance that you get. These wrinkles that are on the surface of each villi are called microvilli. They give you another 20 times the surface as compared to a flat structure. If I take my larger villi down here, I could draw a little bit better down here the fact that now the surface of my villus would also have wrinkles on it. So you've got a wrinkly intestine with little projections that have wrinkles on their surface. If I take 3 times 10 times 20, that gives me 600 times the surface area. And that is why up here, absorption can happen so well in the intestine. All right, we look good. Whoops. So in your small intestine, there are enzymes that are produced there. And I'd really like to emphasize that point. There are enzymes produced by your small intestine. Later on, we'll talk about enzymes produced in a ton of other places, so I don't want you to forget that there are enzymes actually being produced in your intestines. Now, the little video clip that we watched showed this, but I just wanted to make sure you had a good idea of where this was coming from. This is a zoomed-in picture of some villi with microvilli on their surface. This is the zoomed-out picture. What we're trying to get at is that it's coming from right in between each villus. So in between each of the projections, there's a little dip. It's called a crypt. And there are cells there that are making enzymes. So the enzymes are made by cells that line the villi. I'll put the word between here so that hopefully you have a good idea. What I like to think about is, you know that skin that's in between your fingers that it really, really hurts to get a paper cut on? That's where these enzymes are coming from, is the skin or the place between, like let's say where your fingers are attached. The enzymes that are produced there are often called brush border enzymes because, like I mentioned before, the projection or these wrinkles on the villi kind of look like a hairbrush. And there are 4 enzymes that are produced there. There is maltase, which helps digest maltose. There is sucrase that helps digest sucrose. There is lactase that helps digest lactose. And there is peptidase that helps digest polypeptides, which are parts of proteins. So earlier, when we talked about macromolecules, we would have mentioned all of these types of molecules: maltose, sucrose, and lactose. And so I'd just like to remind you what they would be broken down into. For example, if I take maltose, now here's how I know the difference. OSE at the end tells me I have a carb. ASE at the end tells me I have an enzyme. Do all enzymes follow that rule? Unfortunately, no. But if a word ends with ASE, you can be sure that it's an enzyme. So maltase is the enzyme, maltose is the carb. If maltase acts on maltose, maltose is a disaccharide, so it's a thing that's made of two smaller sugars. Maltose is the one that's made out of glucose and glucose. Sucrose, which is the carb, if it's broken down by sucrase, would be broken down into glucose and fructose. Lactase, which acts on lactose, would break it down into glucose and galactose. So remember, all of those disaccharides have glucose as one of the parts of them. It's the second monosaccharide that changes. Does anyone have any questions they'd like to ask about the enzymes that are produced in your small intestine? Yes. You would be expected to know what an enzyme affects and what the result of that is. Now, in some cases, the specific details, like which molecule am I getting, wouldn't be the focus, but since we actually talked about those sugars already, that would be one where I'd expect you to specifically know, okay, maltose is glucose and glucose, sucrose is glucose and fructose. Does anybody else have questions that they'd like to ask about the enzymes that are there? So this is the end of the information about your small intestine. The next thing that we would talk about is your large intestine. And I'll just pause. So your large intestine is divided again into three segments, which I mentioned right at the beginning of this section on the digestive system. The three segments, like these pictures illustrate, let's move this out of our way. The three sections of your large intestine are the ascending colon, the transverse colon, and the descending colon. And so I put a little arrow next to each of them to illustrate to you which direction they're going. So if we take a look just at the outside of your large intestine, that's what this picture is trying to illustrate. Food is going to move through your large intestine this way, up the ascending colon, across the transverse colon, and down the descending colon. So again, in your intestine, large just like in small, food is not moving because of gravity. So in each of the segments, there are muscles, and muscles perform peristalsis. You could assume that basically at every point in your digestive system, a muscle is pushing food to the next point. It's not simply just falling there because of gravity. Now at the end of the descending colon is where we find the rectum. So that was a piece that I identified on the initial drawing of your digestive system. It is a storage organ for feces. We'll call it feces, please, because that is the correct word for it. and it is found immediately after the descending colon. So after the descending colon, you'll see this little U-shaped loop, and then the next little piece is the rectum. Now, just like in your stomach, let's say, there are valves. There's a valve at the end of your stomach controlling the entry of food into your small intestine. There's also a valve in between your small and large intestine. It is called the ileocacal valve. I'm just going to remind you that a valve is a muscle. So just like the valves or the sphincters that you have in your stomach, this valve controls the movement of material from the ileum, which is the third section of your small intestine, into the cecum, which is the pouch at the beginning of your large intestine. And right at the beginning of your large intestine, where all of this is happening, is where you find your appendix. Your appendix has zero actual function in digestion. It might actually be a storage organ for bacteria, but currently, as we know it right now, your appendix doesn't do anything. So I'll pause here.